I'm very honoured to be asked to give this talk. And I would say at the beginning that many of the views I express are my own, not of any former companies. And at the end, I will acknowledge, but I will acknowledge before, because there are many people in the audience who I've worked with in the past, the contribution to some of the data that I'm going to show. I found this lovely quote from the first issue of Nature Drug Discovery. And it was about what a pharmacologist is, what pharmacology is about. And I love the fact that really it's actually also what drug discovery is about as well. And that's one of the things I want to really emphasize is the importance of pharmacology in drug discovery and development, and how perhaps as an industry we haven't always paid enough attention to that. For me, I've loved my time working both in academia, but primarily I've worked much more in industry. And I can't think of a more exciting time to be a pharmacologist, either in academia or industry. The potential is huge, and the question is how best to realise that. Before I talk a bit about some of the projects that I was involved with and some of the lessons I learned, I thought it might be interesting just to reflect for 5 minutes on my background. I was one of 3 girls in the boys' school, the first girls at King's School, so 3 girls, 700 boys. It was interesting. But one of the things that I reflected on subsequently, more recently, is the fact that only 3 girls, there wasn't any cliques, male or female, and I never ever entered my head through the rest of my career why I wouldn't have equal opportunities to my male colleagues. It perhaps gave me a different attitude and confidence that otherwise I might not have had. I actually did physiology and psychology at London University and a PhD on sex and aggression in monkeys, which occasionally, when I sat in all-male groups later, I thought and reflected on some of the sexual displays that I had studied. It certainly gave me a different perspective. I was very privileged to have a Wellcome Trust Fellowship at St George's Hospital in the lab of Cathy Wilson, who sadly has passed away, but she was a wonderful person to work with. And then I decided after 2 years of watching rats copulate in a darkened room, I wanted to do something a little more applied. And I went and applied to work at Glaxo and was accepted to work in the group of Malcolm Caulfield looking at Alzheimer's disease. Interestingly enough, the first day I joined, Donald Strawn said to me, would you rather I treated you as a woman or a scientist? It was odd because I hadn't realized they were mutually exclusive until that point. But I had a great time at Glaxo and moved after a while to Astra, where I worked with Richard Green and Alan Cross, setting up a lab from scratch, which was really a baptism of fire. You reach for your syringes and things, and you don't know where I should have ordered them from, and things like that. But it was a great experience. And then I joined SmithKline & French, which became SmithKline Beecham, which became GlaxoSmithKline. So the moral there is always be kind to people because you never know who you'll be working with again. But it was interesting. I have my daughter and rose through the ranks. Has already been mentioned, as you do become more senior, the number of female faces and actually more diverse faces, actually in terms of ethnicity as well, also decreases. I had a fantastic time at Glaxo. I ran the Neurology and Gastrointestinal Centre of Excellence for Drug Discovery, so that was basic research all the way through to clinical proof of concept. And after 8 years there, I left and set up a company to try and spin assets out of pharma. And we can talk about open innovation at the end because it was an interesting experience, and also, as was alluded to, a consultancy on open innovation. And lastly, at the BBSRC, now the one thing I will say there, I've had a fabulous time at the BBSRC. Today, an action plan for all the research councils has gone to Jo Johnson, Minister for Universities and Science, to really drive change in terms of equality and inclusion across all the research councils. There are targets in there. There are unconscious bias for all the panels. So I am hoping that the research councils as major funders will drive a cultural change, and we are going to be working with Wellcome Trust and the Royal Society to see if there are many more things we can do together. So that's just a plug for that. One of the reasons that I think I'm not going to show the usual doom and gloom about the industry, but one of the things, you might not be able to read this, it's from a review in 2011, that subtly shifted, and actually SmithKline and Beecham only realised it a few years into the merger, was that we moved from an industry that was very much targeted in the main acute diseases to one that was targeting chronic and long-term diseases, and that had implications both in terms of the timelines, the regulations, and also the cost. And that's important because actually the probability of success was less, and actually there was a shift, to lethal, in this term, cancer, etc. Basically, the whole industry moved to a more riskier footing. And you could argue that the current level of attrition is just not sustainable. We, as an industry, the drug company, have to either cut the cost of failure or improve the chance of success. And most of the failure is due to lack of efficacy. Very little in terms of things like pharmacokinetics, which was a major reason of failure in the 1980s. Interestingly enough, if we look at this analysis, there's a big strategic component there, and I think that's something that has perhaps been underestimated. For an industry that has very long-term shareholder gain, timelines, actually there is a lot of chopping and changing, and whether or not that one of the beauties of the SED system was that actually that organisation structure remained for a number of years. So when I went to SmithKline and French, I worked with a number of, in Ray Hill's group, and that's an interesting example of how the BPS can change your life, because I was sitting next to Ray at a dinner at the BPS, and he said, I've got a job going at the Frith, and I said, I didn't know you had a job going at the Frith, and I went along for an interview and I got the job. I hadn't seen the advert, and I wasn't necessarily looking for a job. So networking in that way does pay, just to find out the opportunities. And Roger Egan's group were looking at a compound called Rethinking, which had been being developed out of an antihypertensive program in the States, and it was a selective D2 agonist. And I just show this for those of you who were born after the routine use of cloned receptors, just to sort of show that it was interesting because we were actually looking at the effects of receptors in situ. And it worked the work at, in the lab at SmithKline and French's labs in the Frith showed that actually this compound, which was designed not to get into the brain, clearly did get into the brain and had effects. And the really good thing was that Roger Eden and co. actually thought this could be useful for Parkinson's disease. And they did the testing in rodents, and they worked with Brenda Costel's group to test the molecule in a primate model of Parkinson's, showing here reducing the MPTP-induced motor deficits. So there were two things that are important there. The first is that actually thinking about potential other indications were sort of first, if you like, early repositioning of a preclinical model. But the other thing that I want you to reflect on is the fact that actually this molecule would never have been developed today. Because it had effects on blood pressure, which Roger championed and showed that actually they tolerated out, enabled it to move forward. Today, we do very early ruling of some of the side effects that we see before we've actually looked at the benefit side of the equation. And it worries me. Paracetamol wouldn't be developed today. Aspirin wouldn't be developed today. A lot of very important medications would never have seen the light of day. So it's something we should think about. And the other important thing is the importance of a champion, because Roger championed that. And I remembered that and took it with me when I became SED head. So feed forward 10 or so years, and 2001, the GlaxoSmithKline merger. And in that merger, Tachi Yamada, I think, did a very brave thing. He completely reorganized the organization, and they formed what they called the Centers of Excellence for Drug Discovery. And I think they were very important because they concentrated accountability and responsibility in a group of 300 to 400 people who had, in the past, when new medicines compounds were discovered in the discovery part of an organization, they were delivered to development and they were kind of thrown over the wall. But here there was no place to hide. That was a quote from one of my chemists, because actually you not only had to take that compound, show that it had the developability legs, but also that it had proof of concept. And so we felt very accountable and motivated, and I think it was a fantastic organisation. So the aim was to create something that was more independent and entrepreneurial, and also retain the platform bits of the organisation where scale made sense. And one of the projects that we took forward that had started in SmithKline Beecham, and Carol Routledge was the initial program manager, was to look at the effects of 5-HT6 receptor antagonists. Initially, these had been looked at as antipsychotic agents because the theory had gone that many of the antipsychotic agents that were on the market had very high affinity for the 5-HT6 receptor, especially clozapine. And although a more recent analysis has actually shown that that doesn't correlate with their effects on the cognitive symptoms in schizophrenia, but that was something that's only been published relatively recently. There was a lot of argument, though, that these molecules might be very good for cognitive enhancement. The receptor distribution was appropriate. The antagonists had robust activity in animal models of memory, although the first initial activity was shown in a number of labs, including that of Kevin Fone, using antisense oligonucleotides. And then latterly, there were some indications with effects on neural outgrowth. And the initial lead molecule that came out of SB was 271046. Unfortunately, that had unexpected toxicity issues. It was a very selective molecule, as shown here. And one of the things that was important was we had the knockout mouse at the time, and we were able to show that the toxicity issues were due to the compound and not to the on-target effects at the 5-HT6 receptor. I was actually one of the first people to, in fact, the first person in Glaxo in the late 1980s to use the Morris water maze as a test of memory and learning. And it remains a very popular test. This is just an example of one of the molecule of 046 on the water maze, showing that it increased time spent in the quadrant where the hidden island used to be when the island was removed. Because that compound was not going to move further forward, but we still felt the approach was very important. And one of the things that I would say about 5-HT6 antagonists is that they are incredibly robust in preclinical models of learning and cognition, so much so that we used to use them as a positive control later. The other thing we felt very important in the SED, and we were able to do in parallel, was to always try and develop a PET ligand. Because for me, showing that the engagement of the molecule at the target is really, really important. And in the case of this project, we, Steve Bromwich and co, and Chris Johnson, actually were developing a PET ligand in parallel. This was the lead molecule that eventually made it into the clinic, a very potent compound. Very good oral bioavailability. So really nice developability characteristics. And we used ex vivo binding routinely to sort of set our dose limits as to where we thought we should be going in terms of the learning and memory models. And of course, we were able to reverse scopolamine-induced deficits. Another important finding was that the 5-HT6 antagonists can increase acetylcholine release. Now, a previous speaker talked earlier on this morning about cholinesterase inhibitors. And the dose-limiting effect in Alzheimer's in cholinesterase inhibitors tend to be the peripheral effects. So if you can find a way of elevating central acetylcholine, because these 5-HT6 receptors are pretty much centrally located, without actually stimulating peripheral release, then that's got to be a good thing. And there's also a slight increase in glutamate release as well. Interestingly enough, if you combine cholinesterase inhibitors and 5-HT6 antagonists, this is Aricept with a compound, this is out of a Wyeth patent, you actually see a synergistic effect, which further reinforces the importance of the cholinergic mechanism. And basically, this is just to say a whole range of the first 4 columns here of compounds of 5-HT6 antagonists, and you can see they have very consistent effects on a range of models of learning and memory. So for those of you who aren't familiar with drug discovery and development, this is just kind of the linear process. And so once you have your candidate, you put it into toxicology and safety assays, and then you take it into your first time in human. Now with the 5-HT6 molecules, as I said, we wanted to develop a PET ligand in parallel. We had all clearly the normal information in terms of distribution and blood-brain barrier breakdown from our preclinical species, but did the molecule actually get where we wanted it to go in terms of receptor occupancy centrally? And that is really important, not just for this project, but for other projects, because unless you know that the drug actually made the target, and it made the target in the right concentration, you can never make any conclusion if it doesn't work, whether the mechanism is relevant or not. But if it does reach it at the right concentration and at the right target, then you can make inferences about the mechanism. This was the compound that was selected for development as the PET ligand. It was highly suitable, and if we looked at it, first of all, in pig, and then in human, we could show that the ligand was displaced by the 5-HT6 antagonist, and this is the data in the human brain. So I want to talk a little bit about going back to thinking about Roger Eden. So when we were going to do this study in the SED, this is the phase 2 study, the first study with 457 in Alzheimer patients. And there was a lot of pressure on me to do a small study. I believe the fail fast, fail cheap is just not acceptable. It's not attainable, sorry. It doesn't happen. Now, in the phase 1 data, we'd seen no evidence of any cognitive improvement, although it's hard, but we had no real signals at all. And it would be very easy just to do a single dose and 40 patients, 50 patients per group. But this is where knowing the disease is important, because I knew that if we did that, we would just end up with a completely inconclusive result. But it did mean I had to stand up. I was lucky because I actually, as I said, had held the budget, so it made it a bit easier. I could stand up and say, no, I want to do a big study. And you know what, in my heart of hearts, I actually thought it would fail. But it worked. But you have to have the courage, because I knew if I did the science, if we did, and within the SED, if we did this size of study, we would get a conclusive result. So we did a linear trend design with the 2 lower doses having slightly less power than the top, and the study was powered for the difference between placebo and the top dose. This is what we got with ADAS-Cog, with the top dose showing an effect, and in the global function change, Civic Plus, both doses seem to work. We'd also done a small study looking at Aricept, and that would give us some feeling as to whether we were in the same ballpark as Aricept. And this was important because we had a feeling that the baselines had changed over time with, from when Aricept was first registered. It was registered, you had to have a 3-point change. And if you go back, you can see there wasn't a 3-point change in ADAS-Cog. That was the regulatory requirement. In the small study we did see, although highly variable, we got the similar data between Aricept and 457, so it told us we were kind of in the right ballpark. Now, Lundbeck were developing a 5-HT6 antagonist, and this is data that was published in 2014, showing a similar finding, only this time they were adding it on to donepezil. And thinking about the pharmacology, where you would hope that combining donepezil and the 5-HT6 antagonist preclinically had shown an increase, then we should be able to see still retain benefit and perhaps even more benefit as an add-on therapy. And this molecule has been licensed to Otsuka, and the phase 3 is going. Unfortunately, sadly, GSK did 2 other studies, 2 quite large other studies, one as an add-on and as monotherapy. And although there were some effects, GSK did not feel that they were sufficient enough to move the compound forward, although now it has been acquired and will be looked at in a phase 3 study and also in Lewy body dementia, which personally I think is potentially very exciting because of the effects on dopamine release as well. But I think there's a lot more pharmacology questions that need to be answered about 5-HT6 antagonists. And we knew about some of them, but we just, kind of never had the time, really, to look at them. Some compounds had anorexic effects, and we don't know why that is. Are there any differences between inverse agonists or true antagonists? There are clearly mismatches between the PK and PD of some of the compounds, and we don't really understand why that is. And there are also differences in terms of the effects of agonists and antagonists, sometimes contradictory on neurotransmitter release. There's a lot more pharmacology in 5-HT6, which I think would bear fruit to look at. And this is even more true, I think, in another class of antagonists, the H3 antagonists. And again, we heard something of this morning. Now, the H3 antagonists, personally, I love. I think they're fantastic, and I tried to get my grubby mitts on a few of them when I was trying to spin assets out, but unfortunately, every time we got venture capital, the companies changed their minds, which was sad, but it was very interesting. They work robustly in a number of models. They also increase neurotransmitter release. So H3 antagonists, through their autoreceptor action, will increase histamine. And they also, through heteroreceptors, will increase the release of noradrenaline and 5-HT and dopamine. They have very good brain penetration, and GABA, good brain penetration. One point I will make is that I am very keen on kind of models of mechanism. And so pharmacodynamic assays, when you're screening your compounds on mechanism, they don't have to be related at all to the therapeutic area. And that's exactly what we did here in terms of looking at agonist-induced drinking behavior. So if you give drinking behavior with agonists and then look at the reverse of that with an antagonist, that's a great pharmacodynamic assay. Again, using our favourite water maze, we were able to show that 254 improved water maze performance, and this was the lead histamine antagonist. Pro-cognitive actin in the object recognition task, which is very pleasing. This is where you look at the time spent with a familiar object and then versus a novel object. Actually, interestingly enough, if you give people the same task, they always go for the familiar, but rats always go for the novel. And this is just to demonstrate that in the tuberomillary nucleus, you do see an increase in histamine release with 254, and also in the cortex. So this work, Andy Medhurst and colleagues did a lot of very nice data. Here we did do some work before we went into man, with Mitul Mehta and colleagues, actually showing that you did have effects on fMRI with 294. But interestingly enough, you had to give quite high doses. And this class of histamine antagonists were exquisitely potent. So you were actually giving doses at microgram level. And when we first gave the histamine compounds in the first time in man, actually sort of 0.5 milligrams kept people awake, and were reading newspapers in the middle of the night and things. So we brought the dose down and thought about actually looking at potentially a narcolepsy indication. But one of the things perhaps we didn't pay enough attention to, but reassuringly we weren't alone in the industry, was that actually if you gave the compound chronically and looked at, in this case, in orexin knockout mice, the effects actually did reduce after 10 days of dosing. And when we studied this molecule in narcolepsy, we didn't see an effect in the small study that we did. A sister molecule, 512, was developed in Alzheimer's disease and was tested in Alzheimer's disease, and unfortunately did not have an effect. In both the narcolepsy studies and the Alzheimer's study, the molecule was titrated up to reduce unwanted effects on sleep, too much effects on sleep. And I think What we failed to realize perhaps is that this is agonist pharmacology, not actually antagonist pharmacology, because you are mediating the effects through the release of histamine and other neurotransmitters. And I think doing this actually highlighted the need to really think very carefully about where you need to be on the dose-response curve and receptor occupancy curve. And the molecule also had no effect in a schizophrenia trial. We weren't alone. The same with the Merck compound. Again, they're very small patient numbers, but no effect. Now, interestingly enough, pitolisant is an H3 antagonist that is, it's not approved, it's recommended for approval for the EMA just last month. And this is pitolisant in a trial comparing it to modafinil, which is the standard agent in narcolepsy. And you can see very nicely that in terms of the sleep score, you get the same effect on the reduction of ESS score, excessive daytime sleepiness score, as modafinil with pitolisant versus placebo. And so this molecule clearly is different in that you can see effects on sleep. Again, going back to the MK compound in sleep, again, acutely saw an effect, chronically did not see an effect. So these are things we should be thinking about in terms of looking at the evidence for the pharmacology and should one go down in dose. There are few differences between these molecules, although the protein binding is much higher with pitolisant, which might suggest that actually the exposures in the brain are less and reinforce the need for lower dose pharmacology. So I think with the histamine receptors, there are a lot of unanswered questions. Some histamine antagonists are effective as anti-epileptics. Some have effects in chronic pain. We do not know why, what are the differences for those particular, between those particular compounds. Is it there are differences at the heteroreceptors? And I do think we have to think very carefully about this fixation about pushing everything up to the maximum dose. So my lessons from studies on cognition at the sort of coal face in drug discovery, there's a lot of variability. So trying to actually find phase 1 markers of cognition is very hard. We should be going for mechanistic, wherever possible, mechanistic kind of outputs. And this, phase 2 studies are variable, and we need to make sure we power them appropriately. We really need to be very clear about our PK-PD relationships and using PET to actually define the degree of receptor occupancy that you need, and it might not be full receptor occupancy is important. And so, the mechanistic pharmacology in terms of informing clinical trial design is really important. So now I want to sort of move away and think a little bit about the future of pharmacology. Because I think we have to embrace a new way of thinking and doing things. Because we're moving from a model where we had the human disease, we did some in vitro biology, we had our in vivo models of disease, and then if we got a positive in our in vivo models of disease, we would move onwards. We're getting incredibly important data now from human genetic studies, and we need to find ways of moving much more swiftly from those genetic studies. Now, a few years ago, I was asked by Science Translational Medicine to write an article about animal models of mechanism, and it didn't get published because the furry rodent disease model people, I think, were a bit upset by it. And I speak as a furry disease model person myself. I mean, I was a behavioral pharmacologist, but I really think we place far too much over-reliance on animal models of disease. Many of them are animal models of mechanism. The transgenic mouse is an animal model of amyloid deposition. It's not a model of Alzheimer's disease. And we are also finding, I think, as scientists, that we are getting ourselves into problems in terms of this reproducibility question. Because when you get to a situation where at least 2 drug companies did a whole range of studies and couldn't replicate 80% of the findings that had been published in academia, it's very worrying. If you control your models so much that you see things that are statistically significant but are not biologically significant, then I think, the inferences we can draw from those models have to be limited, especially if you're going to talk. That phase 2A Alzheimer trial I showed you with the 3 doses cost £6.8 million. And, we can't go into these studies lightly. They are, and we don't want to expose patients to agents that are not going to do any good. Recently, the BBSRC funded some work looking at stress and the effects of stress on the behaviour of offspring. Clear differences between males and females in the output. I know why we use males, because it reduces variability. But we also have to think about whether or not this homogeneity in terms of how we try and corral the experiments actually is something that is giving us valuable data. Of course, as funders, we also have to look at the implications of that, because it will increase the numbers of animals you have to use. I think it's very important that we try and get some standardizations of methods. It's really, it's like somebody said, getting a scientist to share their data is like getting them, they'd rather share a toothpaste, but I think, a toothbrush. But I think that initiatives like the IMI have begun to, at least at an industry level and to some extent academia, agree on certain standards for testing. Now, this was done in the 1990s for stroke, and the consortium that came out of that agreed on protocols for testing for stroke. Companies and academics use the same protocol, and what has been fascinating about that is that because that data is available and it's all collected in a very standard way, recently there was a meta-analysis done of all that data using new techniques, which enabled researchers to pull out some new important information about variables that would predict poor stroke outcome. And so I think that, that's really important going forward. The other reason why I'm pro-animal models and mechanism is it avoids you having to test your molecule in the gazillions of models that everybody has their own pet model. This is just a real-life example of a compound that was tested in 3 different animal models of neuropathic pain. On the left, you can see the actual doses, exposures that were effective at those various models. And on the right, you can see the plasma concentrations of drug from the first time in man study. So which dose do we test? Well, we did what we always do, which is kind of go up to the highest tolerated dose. But again, this was an agonist therapy. Was that appropriate? So I believe that we really need to think differently about how we do drug discovery and development. And I'll show you some data in a moment that will support that we need to move more to animal models of mechanism and actually focus on developing those to enable us to be able to translate from mechanistic in vitro, mechanistic in vivo preclinically, and mechanistic in vivo in man, in our first time in man. I think that will reduce our attrition, because we will be able to identify those compounds that don't get at the target earlier, and also allow comparison with the pharmacodynamics and the pharmacokinetics. And it does require a different mindset. And if you're in a drug company and you're taking your molecule forward and you have your science advisory board, I can tell you I've been in a room where I've had several advisors all telling me to test something in a different model, and they won't agree which model to test is the right one to test. And that's not really helping patients, I don't think. And so we have to think about a new paradigm which will accelerate the use of this genetic knowledge into in vivo models of mechanism and more rapidly into the clinic. And some of this has already been proven. If we look at the neuropathic pain at the sodium channel variants, we've almost got in vivo human target validation. And molecules out of that stable have proved to, or are looking promising in the clinic. So, we need to be able to capitalize on this. We need to be able to capitalize on the ability of us to be ourselves to be able to take tissue from patients, to be able to remodel pluripotent stem cells for the tissue of interest. And there we're looking at our target in the right environmental milieu. Because we know you can have a mutation in a particular ion channel. If you express that, which is causal in, say, epilepsy, if you express that in a cloned receptor, a cloned system, in an artificial system, you don't see any change in pharmacology, but you do if you then take the stem cells from the patient. And we know this must also be the case because we have a lot of genes that we know are susceptibility factors that individually are not causal, but together, in concert, you will see an effect. And I think that requires a new business model, a new way of working. Public-private partnerships are, public should say PPPs, not PPIs. I think I was involved in the beginning of setting up the Innovative Medicines Initiative, and I think the Innovative Medicines Initiative has shown, certainly IMI1, that companies and academics can work together very effectively to share information and to share best practice. So, for example, in the schizophrenia project, actually coming up with a standard design for a schizophrenia trial with the endpoints that were agreed, which meant it reduced time and costs and numbers of patients. So there's real benefit in pre-competitive collaboration. I don't see quite so much in academia, but that's often because the system rewards individuals rather than collaboration across institutions, although that is clearly changing. And thinking more laterally and exploring the boundaries for how we can really be more innovative in drug discovery and development. Sorry, going back. I mentioned that I was going to show something about the mechanistic importance of mechanism, and this is data that was published this year from Chorus, which is kind of Lilly's sort of internal spin-out. And basically, Chorus takes molecules with the particular developmental characteristics listed on the left. They've got biomarkers, small patient cohorts, focused indication, etc. So it's quite a limited subset of compounds that Chorus takes out for fast development. But what I thought was interesting from this graph here is that if you look at the 41 programs over the period, the 5 years they're looking at, 5 exited after toxicology, which is about kind of what we would see, I think, across GSK, drug development and discovery. 17 exits after proof of mechanism, 9 after proof of concept. So nearly half were weaned out by that proof of mechanism. Very interesting data, I thought. And the other thing I think about pharmacology is, I was talking to somebody yesterday in the industry, and they said, actually, there's a dearth of pharmacologists in the US, and we've had to recruit people from outside the US. I don't think we've paid enough attention to pharmacology as an industry. I think we've been a bit seduced by molecular biology. And pharmacology is at its heart, it's a systems approach. And it's a systems approach that we need going forward. And we also need to listen to the evidence, though. So when we do the pharmacology, actually listening to it, I remember in stroke, the pharmacology, the time course in the animals tells us compounds don't work after 2 hours of post-stroke administration. And it's the same with the thrombolytics. But because there was seen to be no commercial market unless a compound worked at 6 hours, compounds were tested at 6 hours. There was actually, the evidence didn't support that. I think pharmacology has a huge role to play going forward in this mechanistic study and actually being able to embrace biomarkers and biologicals. I see bispecific antibodies, the pharmacological questions in terms of on rates, off rates, relative distribution, etc., are really, really important. And we heard earlier again this morning about precision medicine. I think if we're going to move to an era of precision medicine, we need to move to an era of precision pharmacology. So I think that pharmacology. I was so pleased the other day. I was in the hairdressers, and the person who I've known for a long time there, her daughter was helping out. And I said, what do you want to do? She said, I want to be a pharmacologist. And I was like, wow, that's really good. She said, yes, the careers advice person at school didn't know what it was. She said, oh, well, first of all, she said to this girl, I don't think, you should aim a little lower, because she hadn't seen the grades this girl was going to get. And then when she saw them, she said, oh, well, yeah, okay, perhaps you could do it. But she didn't know what it was. And I think that's really sad. So we've got an education job to do. I hope that all of you who are starting out in pharmacology will continue in pharmacology, because I think if we don't if we don't use pharmacology to enable the knowledge coming out of genetics and genomics to create, for me, obviously there's knowledge, but also new and valuable medicines for people, we are doing a disservice, really, to science. So, you know, I think the future is bright. I'd like to acknowledge all my colleagues that I've worked with. It's all their work, and we're all fantastic people. And also my family and friends, because they always bring me down to earth and keep my life in balance. Thank you.